Gene Cloning by Omar Habashi. Human cloning has been an ethical question ever since scientists began theorizing about it. Religious beliefs regarding what humans should do in science have heavily influenced governments' actions, as in the past no one was open-minded. In the early 1900s, the human population was 1.62 billion, with 1,000 religions dominating the population and an approximate number of 1.3 billion religious people globally. Past ethical issues such as normalizing plastic surgery, sexual preference, and/or orientation have been widely debated. However, today, ethical committee and boards have become more lenient towards preference. This allows individuals to choose the person they want to be with no ethical or religious barriers between them. With more and more scientific and biotechnological ethical issues rising as the speed of science discovery skyrockets, there is only one ethical question that has not yet been answered. That question is, should we be able to clone our genes? In 1885, a German scientist known as Hans Adolf Udard began researching production. Adolf's research resulted in the creation of the first ever study about cloning. He was able to separate two viable salamander embryos from one, creating the concept we know today as gene cloning. More than 100 years later, the most famous case of gene cloning to ever exist took place in Scotland, where the first cloned mammal was born. The sheep named Dolly was a revolutionary benchmark for gene cloning. The remarkable discovery of how one adult cell can be used to create an entire organism faded quickly after the death of Dolly. In 2003, Dolly died at the young age of 6 due to osteoarthritis and lung infection. Dolly's death sparked new questions. Are cloned organisms more prone to disease? Do cloned organisms age at a faster rate than normal organisms? And why did Dolly die young? These questions have not been answered, as no one today is cloning full organisms, but are in fact only cloning small pieces of DNA such as genes. There are three main steps to gene cloning. Let's imagine we're trying to clone a gene. Step 1 is cutting open the plasmid and placing a gene copy into it. The plasmid can be identified as the region where DNA fragments and/or strands can be melted to begin the replication process. A plasmid contains three elements. The first element is cloning site, where foreign DNA fragments can be inserted. The second element is a drug-resistant gene, or AMP to the power of R. The third element is the replication origin. It allows the plasmid to replicate in the host cell. Step 2 of the gene cloning process is preparing the vector in which the genetic material will be placed inside. Step 3 of gene cloning is introducing a restriction enzyme that will allow the cloning site to cleave the vector. An example of this enzyme is EcoR1, which cleaves the cloning site, creating single-stranded ends in the process. These ends are referred to as sticky ends. The enzyme, in this case EcoR1, digests the DNA fragment that is wished to be cloned and is inherited by the sticky ends of the single-stranded DNA of the plasmid. Both the DNA fragments and the sticky ends hybridize and seal with phosphodiester linkages. Enzymes that help with DNA replication, such as DNA ligase, aid when sealing this linkage. This creates a recombinant plasmid. Cells are then added, to which only a few plasmids enter certain cells. This process is called transformation. The cells are placed onto agar plates of nutrient-rich chemicals, where they will stay in the most effective environment for growth. Plasmids will use resources and enzymes from the cell to replicate. When cells replicate, each daughter cell will contain a copy of the plasmid. The result is a colony of recombinant cells containing the foreign DNA fragment that was first inserted. Some advantages are a great contribution to organ placement, will allow us to venture the land of mystery, revolutionary scientific benchmark, provide cures for some diseases, it could extend human life capabilities, biological children could be created for same-gender couples, allows infertile couples to reproduce. Some of the disadvantages in gene cloning is the uncertainty of viability, might bring new diseases that we are not ready to fight, will most likely lead to further destruction of the environment, risk of being abused by commercial farms, labs, etc., will cause a lot of negative backlash, will always be trial and error, could create organisms who will only live their short lives to suffer until their death, and organisms will be tortured in labs for the sake of experiment. In Canada, the 2004 Assisted Human Reproduction Act bans any and all activity relating to human cloning and states that such actions are criminally prohibited. Many countries such as Australia, Austria, Argentina, Belgium, Brazil, Czech Republic, Costa Rica, Denmark, France, Germany, Italy, India, Japan, Lithuania, Mexico, the Netherlands, Norway, Peru, Portugal, Romania, Russia, Slovakia, South Africa, South Korea, Spain, Sweden, Switzerland, Tobago, the United Kingdom have explicit laws banning human cloning of all sorts, ranging from single cells to full-grown organs. Religions including Islam, Christianity, Hinduism, Buddhism, and Sikhism all prohibit any and all actions remotely related to humans creating life in the laboratory environment. This is because these religions believe that only God or gods are allowed to create life. Personally, I believe it is unethical to clone genes, offspring, and babies, human or animal, for the sake of research and/or scientific discoveries. I believe that only God has the power to create life and that science should focus on how to use medicine to solve existing problems rather than create problems for our future generations. Others may disagree with me, and that's completely fine with me, as I also believe that people should be entitled to their own educated opinions. Some may argue that cloning is a way to reduce human suffering from diseases relating to organs, as cloning in the future is predicted to be able to clone full viable organs. However, I would argue that the uncertainty of the future outweighs the benefits correlating to cloning. In our first unit, we learned about biochemistry. We watched and analyzed a documentary about how life was first created here on Earth. The documentary stated that it is believed through science that life here on Earth was created in the oceans by non-living things. This raised the same question I have discussed today, the question of beliefs and the creation of life. The end. Thank you for watching. This is Omar Habashi. All my references are in my transcript, and have a nice day.